back to Science of Medicine. A little while ago, I briefly touched on some highlights of what the kidneys do in renal physiology. Since we're going to be talking about a lot of chemistry across this next block of classes, I wanted to go into detail on renal physiology as it's very pertinent to a lot of the chemical applications in the body. So to remind you, your kidneys do a lot of things. One of the first things that they're responsible for is the maintenance of this homeostatic fluid compartments, that is the interstitium in which cells live, to make sure it's got the right amount of water, electrolytes. In order to do this, I have to remove waste that is generated in the body through such as breakdown of hemoglobin or in the normal metabolic processes of protein degradation. And I need to reclaim substances that I've spent energy to actually make or absorb, things like glucose and amino acids. The kidneys are also responsible for making several things. The two things that are most pertinent to our class are erythropoietin, which is the hormone that stimulates red blood cell production in the bone marrow, and the activation of vitamin D. Now, vitamin D can either be absorbed in the duodenum or synthesized in the skin, but in order for it to be activated, I have to add a hydroxyl group to it at a particular place on that steroid backbone. And so that is done in the kidneys. There's also regulation of acid-base balance, particularly with regard to acids that cannot be ventilated through the pulmonary system, and we'll talk in great detail about acid-base chemistry. And then the other thing that the kidneys are responsible for is regulation of arterial blood pressure, both through the renin system as well as through sodium and water regulation. Now, if I look at kidneys, most of us will have two kidneys, although sometimes some people will only have one. You only need one for survival. And sometimes people are only born with one kidney. And those kidneys, if you have two, will lie underneath your abdomen on the back side in what's called the retroperitoneal space. And their purpose is by maintaining the homeostasis inside. In order to do that, they have to filter blood. So they need a good blood supply. So the renal artery is going to come off of the abdominal aorta and go to the kidneys. And it will then break into smaller and smaller blood vessels and then eventually go into what's called the glomerulus, which will then be part of about 1 million nephrons that you have in each kidney that will cause all of these things to happen, and we'll talk about in detail. Now, if I look at the renal, the kidney on its side, I find that the outside is made up of cortex, this fibrous capsule is even beyond that. And then I have an inner portion, which is the medulla, and then I'll eventually collect all of the filtrate or the urine that I'm making in these calyces, which will go down my ureter into my bladder. The filtered blood will come back into the body through the renal vein, and I will then actually be able to then take that purified or filtered blood back into systemic circulation. So the basic functional units, the thing that carries out what the kidney is doing, is the nephron. And as I mentioned, there are about 1 million of these in fully functional kidneys, and that's made up of several parts. So first off, as I mentioned, the renal artery coming off the aorta will actually then get to smaller and smaller vessels, and eventually they'll go into this glomerulus. So the input into that glomerulus is called the afferent arteriole. And the outside, the one that is going to go back into the venous system, is going to end up being the efferent arteriole. And I have a whole system around the nephrons of what are called peritubular capillaries that are going to allow me to reabsorb things that are going to go on in the kidneys. Now, that first part of the kidney, as I mentioned, was the glomerulus. After the filtrate goes into Bowman's capsule in the glomerulus, it will go into the proximal convoluted tubule. And in this section, I'm going to have mostly reabsorption of needed materials, as well as secretion of some unwanted things. It's then going to go through the loop of Henle. Now, the only purpose of the loop of Henle that we're concerned with has absolutely nothing to do with the filtrate. It actually has to do with the creation of an osmotic gradient as I go down this filtrate, and as I go down this loop of Henle. And that's going to allow for my cortical collecting duct to either concentrate urine or not concentrate urine, depending upon some additional information, which we'll talk about. I then go into the distal convoluted tubule, where I'm going to do basically just a few things. I'm going to be able to adjust the flow in the glomerulus. I'm also going to be able to deal with both potassium and hydrogen ion regulation through that distal convoluted tubule. And then eventually I'll go into the cortical collecting duct, where I will either reabsorb water or not reabsorb water and take that urine down into the calyx, which will then go into the bladder. All right, so let's spend a lot of time actually talking about the glomerulus. So the glomerulus is made up of a couple parts, and it's actually important to understand these parts to understand what's going on. As I mentioned, I have an afferent arteriole that is coming previously off of the renal artery, although it's broken down into many, many smaller blood vessels before it gets to the glomerulus. And this tuft of capillaries will then go into this Bowman's capsule and then go out through the efferent arteriole. Now, a lot of things need to go on in this glomerulus to allow for filtration of blood, because that's my primary mover here, is I'm trying to filter the blood. I don't care about making urine. Urine is cheap, right? Urine is a waste product. No one goes around collecting urine for the sake of urine. I need to be able to take what's waste, get rid of it, and keep what's good. And that's this filtration process. So if I look at a picture of the glomerulus in side view, I would find that these capillaries would be coming in and out of this picture. And these capillaries have endothelial cells, just like any other capillaries. And surrounding those glomerular capillaries are these what are called podocytes, which are cells that have these feet-like projection that envelop the glomerulus, or these capillaries. And in between the glomeruli, the glomerular capillaries, there's slit-like spaces that is going to allow for a level of filtration. In order for filtrate to go into Bowman's space, which is shown here, and then eventually go into the proximal convoluted tubule, the filtrate has to make it from the glomerular capillary into Bowman's space. And in order to do that, it's got to go through a variety of things. It's got to go through those endothelial layers, which are a layer which is thick. It's got to go through these podocytes. The podocytes also have a basement membrane that is between it and this space. And then and only then it will be able to get through and go into the proximal convoluted tubule. There's also sort of a wastebasket, which is in the mesangium, which is very important, especially for kidney diseases. There are a lot of cells that are down in here which can cause problems. As you can imagine, with any filtration system, the filter can sometimes get clogged, and so you have cells down here that will basically go and clean this, this filtration apparatus. Very, very important. There are also an enormous amount of diseases that happen in the glomerulus. These diseases can be, as you can imagine, things that involve this filtration process. When you gunk up or clog up this filtration apparatus, there are a lot of problems, and you can deposit things in this area that leads to both an immune response within this, or as well as just simply clogging this up. And lots of human diseases are associated with this. Now, I also will point out, but come back a little bit later on and talk about it, I also have juxtaposition to the glomerulus, a portion of the distal convoluted tubule, and that's going to be a sensor for filtration through here. That's the maculodensa and also this juxtaglomerular apparatus. And I'll come back when we talk about that in the distal convoluted tubule. Now, for the most part, whatever makes it into Bowman's space and then into the proximal convoluted tubule, if this process is working correctly, is devoid of cells and proteins. For the easiest way of actually thinking about this, pretty much you filter only things that have an atomic mass unit of about 70,000 atomic mass units. So albumin is not filtered into Bowman's space. It is actually not in the filtrate. It remains in the capillary. Red blood cells, white blood cells, large cells like that, they never make it through if the filter is working properly. And there are, like I said, many diseases where that is not the case. And when that happens, then you can actually have things that get into the urine that shouldn't be there. Things like cells or proteins. Now, this is a microscopic view of this tuft of capillaries in the glomerulus. And you can see these striations of those podocyte feet that are actually enveloping these capillaries. And as I mentioned, this is one of the layers of filtration that you actually have. And underneath those, on even higher magnification, this is now 200 nanometers, so much, much smaller than what you saw before, is this fenestrated basement membrane. And this fenestrated basement membrane has these holes through which you can also pass filtrate into Bowman's space or Bowman's capsule. The basement membrane is made up of proteoglycans that are negatively charged, and so you not only get a size, but you also get a charge filtration. Although one of the predominant ones is really in terms of the size. Now, as I mentioned, the purpose of this glomerulus is to filter blood. It's not to make urine. Again, I don't care about the urine, I care about the blood being filtered. So the blood comes in through the afferent arteriole, this filtration process goes through, and you're left with the filtrate, which then will be eventually modified and excreted. What's left coming out the efferent arteriole, though, are going to be a more purified portion of blood, and then I can also reabsorb things that I want to actually restore back to the bloodstream, things like glucose or amino acids, as well as I can secrete things from there into the proximal convoluted tubule that I want to get rid of. Now, the rate at which I'm filtering depends upon what this filtration rate is, what the reabsorption rate is over here, and what the secretion rate is. And I'm going to talk about this, what's called glomerular filtration rate, in just a second. So, in mathematical terms, that filtration is dependent upon a couple factors. The first is the difference between the hydrostatic pressure inside the glomerulus and Bowman's capsule. And as you can imagine, the higher the hydrostatic pressure inside those capillaries and the lower or the lower pressure within Bowman's space will lead to more filtration of fluid by virtue of hydrostatic concerns. The other thing it depends upon is the oncotic pressure. Now, if I'm going to be filtering fluid but not filtering things like albumin, albumin will exert an oncotic pressure that will tend to hold fluid in as you go down the length of the glomerulus. So early on, the hydrostatic pressure is sort of the dominant term in this. And later on, the oncotic pressure becomes very important because I'm concentrating the albumin by moving out fluid. I also have a filtration coefficient, sort of an empiric factor related to the leakiness of this capillary. Now, I mentioned before about regulating, and this is the process by which the kidney regulates this filtration is called autoregulation. And in order to do that, I need a sensor. And I mentioned to you before that this sensor system is located in the distal convoluted tubule. And we'll talk about later on that we have different types of cells like principal and intercalated cells in the distal convoluted tubule that will be involved in hydrogen and potassium regulation. But now I'm talking about a special set of cells in the macula densa that are going to sense the delivery of sodium chloride to that distal convoluted tubule. Now remember that filtrate is going from Bowman's capsule, proximal convoluted tubule, loop of Henle, into the distal convoluted tubule. So if I filter less, it must mean that I have less filtrate that's getting to the distal convoluted tubule, and the macula densa can actually sense that. And if it does sense that, then it goes, oh, I must not be filtering enough through the glomerulus. And so I'm going to release renin. Now you may remember renin is a hormone that will then convert angiotensin-1, lead to its conversion into angiotensin-2, and it turns out that that is a constrictor of the efferent arteriole. So if I think in very simple terms, as a tube going into this glomerulus and then another tube coming out, remember Poiseuille's law shows us that if we have laminar flow, that the flow through a tube is dependent to the fourth power of the radius of that tube. So if I look at this, if I want to adjust the flow, I can either decrease the flow by decreasing the radius of the afferent arteriole or increasing the radius of the efferent arteriole, or I could do the opposite. I could increase the flow by either increasing the afferent arteriole's radius or decreasing the efferent arteriole's radius. And so this feedback mechanism has to have a sensor system, and it has to have a way chemically of doing this. And it does this through this constriction and relaxation of these afferent and efferent arterioles with different molecules. I mentioned that angiotensin-2 is a constrictor of the efferent arteriole. If I constrict the efferent arteriole, it creates an increase in pressure, which will then allow for greater filtration in the glomerulus. I also have some other vasoconstrictors. I have epinephrine and norepinephrine, and then also endothelin, which is a potent constrictor, and it can be either afferent or efferent arterioles that actually express those receptors for that lead to constriction. I can also have dilation or an increase in the radius. Nitric oxide, NO, actually dilates both the afferent and the efferent arterioles, as well as prostaglandins. And prostaglandins are dilators. Now, prostaglandins are a series of a type of molecule that are created in the cell, and you can block this by the use of non-steroidal anti-inflammatory drugs. Things like Aleve, ibuprofen, aspirin can block this. So as I remind you again, if the macula densa senses a hypoperfusion, then it will allow for renin to be released. Renin will go up and then allow for the conversion of angiotensinogen into angiotensin-1, which with the ACE, the angiotensin converting enzyme created in the lungs, will go to angiotensin-2, which can then vasoconstrict the efferent arteriole. It will also do other kinds of things, such as allow for ADH to be released so that I can reabsorb water. But it will ultimately, in terms of its effect on GFR, glomerular filtration rate, constrict the efferent arteriole, and then also allow for increased water and therefore fluid and therefore pressure. So this is the kidney's adaptation to hypoperfusion. Now I've gone through the glomerulus, and we've talked about how you filter the blood, and you're left with this filtrate, which is going to eventually become urine. Now the first part that it's going to go through is the proximal convoluted tubule. Proximal because it's closest to the glomerulus, convoluted because it goes back and forth in a convoluted fashion. And it is where you're primarily responsible for a reabsorption of nutrients from the filtrate and secretion of sort of unwanted molecules in the blood. Now, that means that if I spent a lot of energy in absorbing, let's say, glucose or amino acids that are in the from the gut, I don't want to just pee them out. I don't want to get rid of those. So I have transporters that will allow me to reabsorb in the proximal convoluted tubule those things that I want. Now, this is another very detailed picture of this proximal convoluted tubule over here. And what you see in terms of this is that in order to actually have that reabsorption, I have to have several things that actually occur. First off, in the proximal convoluted tubule here, I have to have transporters that will either specifically absorb a particular substance or generically absorb a class of substances like organic acids or something like that. I have to have that facing inward to the proximal convoluted tubule so that it can actually pick up those substances. I then need to be able to pump those substances or move them outside the backside of the proximal convoluted tubule into the interstitium, and then they need to be picked up in this capillary network that's going to then go back into the bloodstream. So I have several processes that need to go through that. Many of those are energy requiring processes. Some things are passively transported, such as water and urea and sodium chloride, but most of them are actively. They need ATP. So things like glucose and amino acids, as well as things like lactate and phosphate and sulfate. And in fact, this is really important. There is a class of drugs called the flozins that actually are very, very effective at blocking the transporter in the proximal convoluted tubule for glucose. And people with diabetes actually will, if the glucose starts to get above about 180 milligrams per deciliter, they'll saturate these transporters, and so they'll start to pee out glucose. It's one of the problems that diabetics will have. And when that happens, if you block the what's called the sGLUT2 proximal convoluted tubule receptor, then you'll pee out more glucose and you'll have less glucose in your blood. It's one of the ways that we actually will help for better control of a person's diabetes mellitus. Now, there are a few substances that I will also have receptors to secrete into this. Catecholamines, so if somebody scares you or startles you and you have adrenaline or norepinephrine, I need to get rid of that in a fairly fast period of time because you don't want to be in that hyperadrenergic state for hours after somebody's startled you. And so it will secrete it in there. Oxalate or bile salts also get secreted into the proximal convoluted tubule. And as I mentioned to you, in order to get this to happen, I have to have transporters on both sides of the membrane, one that would be facing into the lumen of the proximal convoluted tubule and the other out the basolateral membrane. Some things, like for some regulation of acid-base balance, I actually create molecules that will then create acids, and I can pump an acid in one direction or bicarbonate out in another direction. Once I've gone through the proximal convoluted tubule, I then go into the loop of Henle. If I look at the filtrate at the beginning of the loop of Henle and the end of the loop of Henle, it's the same. There's no difference in its chemical composition. So you go, okay, well, why do I have this large dip down in from the cortex of the kidney into the medulla? Why not just bypass it and go here? And the reason for that is that I have to create an osmotic gradient that is in the interstitium adjacent to the cortical collecting duct. And the reason that I need to do that is that if I need to reabsorb water, I have to have a driving force for doing that. And that driving force is going to be the osmolar gradient. If I look at the osmolarity up here close to the cortex at the very top of the loop of Henle, it is much, much less, somewhere around, let's say, 300 milliosms per liter, compared to the bottom, which can go down to as high as 1,200 milliosms per liter. And that's a fourfold concentration difference, which allows my interstitium around the cortical collecting duct to be about 4 times concentrated power for water. And that's very, very important. But in order to do this, I have to have active processes that go on to create this osmolar gradient. Now, the osmolar gradient is comprised of sodium chloride as well as urea. I'll just touch upon the sodium chloride, which is used through this active transporters, which are located in the ascending loop of Henle, which pump out sodium potassium chloride into the interstitium. There's another process that leads to a urea gradient. But the key idea here is that I need to be able to create this gradient in order to concentrate the urine and reabsorb water in the cortical collecting duct. And that's the purpose of my loop of Henle. Otherwise, I won't be able to reabsorb passively water as I do this. For those of you who have heard of things similar to this, this kind of what we call countercurrent mechanism is similar to what fish use to extract water in their gills. Also an old-fashioned term, Craig counterchromatography. Back before we had liquid chromatographs, we could do multiple extractions, and that same process is the way in which the loop of Henle allows for this concentrated interstitium. Now, once I've gone through the loop of Henle, I go up to the distal convoluted tubule. As I mentioned, part of the distal convoluted tubule has that macula densa, which will be essential for me doing the autoregulatory part of my GFR. But I also have two other types of cells. I have principal cells and intercalated cells between them. The principal and intercalated cells both respond to aldosterone. Aldosterone is a hormone that will actually then facilitate many things, but one of them is that it will act on the principal and intercalated cells. If aldosterone is present in the principal cells, it will increase sodium reabsorption and release potassium into the interstitium, into the filtrate, which means into the urine. So by doing this, if I have aldosterone, I will decrease potassium inside my body, in the blood, and increase it in the urine. In other words, it's a way of wasting potassium. Now, I may want to waste potassium because potassium is very high, or I may end up wasting potassium because sodium is very essential to me to maintain my volume. And so this is done under aldosterone, principal cells. I also have intercalated cells. Now, intercalated cells are also under the influence of aldosterone. And if aldosterone is present, I will pump a proton into the urine, and that will then allow for an exchange of bicarbonate with chloride in the renal interstitium. So I can adjust in a very slow fashion, but I can adjust the pH in my body and in my blood via this renal mechanism. It's a much slower mechanism than, let's say, if I ventilate carbon dioxide in my lungs, but we'll talk about that in more detail when we talk about acid-base chemistry in the body. Now, I can also, I alluded to in the proximal convoluted tubule, I can also adjust my bicarbonate through the reabsorption of bicarbonate or the generation of bicarbonate. And this happens when you eat a lot of meat. Proteins from meat sources are acidic. They will end up generating ammonia, which will then end up yielding an acidotic condition. And so that ammonium ion is actually is going to create a pH imbalance, which I will then be able to adjust for. And so in the body, this is mediated in the proximal convoluted tubule with glutamine, and in the distal convoluted tubule through this aldosterone-mediated process. So what I want you to remember is that if I have aldosterone, if aldosterone is present in a large amount, I will not only reabsorb sodium and kick out potassium in the urine via the principal cells, but I will also pump hydrogen ions into the urine and therefore create a less acidotic blood. And that's important for what we'll talk about a little bit later on. Now, once I've made it through the distal convoluted tubule, the only thing left that I can do in the cortical collecting duct is deal with water. And that's what my cortical collecting duct is doing. It is going to either absorb water or reabsorb water if it needs to, or not. And that's it. Otherwise, this stuff goes down into the ureter and then goes out into the bladder and you pee it out through your urethra. The molecule that is controlling this is called ADH, or antidiuretic hormone, or vasopressin. This is a molecule that is released from the posterior pituitary gland. And if I have antidiuretic hormone present, the cells in the cortical collecting duct we'll insert what's called an aquaporin channel, which will then allow for the reabsorption of water into the body. There's a peritubular capillary network here, which will reabsorb it once it goes into the interstitium and then go back into the blood. Remember that that blood is more concentrated because I've filtered out a lot of fluid, and it's got albumin, which I haven't filtered out. And so with this ADH or antidiuretic hormone, I'll insert the aquaporin channels. If those aquaporin channels are not present, or if the osmolar gradient is not present in the interstitium, no water is reabsorbed. That is necessary, or I have to have both. So this has been an introduction to more details of renal physiology. We're going to use this more in the next segment when we talk about acid-base chemistry. And I'll see you then.